My name is Joachim R de Miranda, and I work at SLU, not together with Ricardo, but in the same institute, as a researcher in the honeybee pathology group. And first of all, like everybody else, I would like to thank the organizers and Jack Kislanek in particular for inviting me here to give a little talk about honeybees and their diseases. It's important that we at the universities share our work with the public that in the end supports us for doing our work, and with the beekeepers and the authorities. And it's also very encouraging that the public and the beekeepers are interested in our work. So these type of events are super interesting for us as well, and I think super important. I would also like to thank both of the translators who've kept us, at least me, in tune with all the other talks as well. It went a little too fast for us in Swedish. So the title of the talk is Diseases, Mites and Viruses, Impacts and Solutions. And the subject that I was tasked to talk on, it's a very broad topic. And so I will split it into three parts. First of all, we talk a little bit about the diseases. What is killing bee colonies? Then the impacts, how are we managing with these? And then finally, looking into the future a little bit at solutions. Briefly about the solutions already available, and also about things that we're thinking about. So, first of all, the diseases. There are many organisms that call the hive their home. It's not just honeybees. We have pathogens. There are a number of them: viruses, bacteria, spiroplasmas, protozoa, and fungi. In this talk, I will be concentrating on the viruses here. There are two very important bacterial diseases as well. Parasites, Microsporidian nematodes, etc. Here we'll be taking a look at mites in closer detail. And then predators. These are a very interesting group and colorful that live on the bees themselves. And then, of course, in the hive, there are microbes, bacteria and fungi that eat the products, that live off the products that bees produce, both pollen and honey, and also the wax and even the wood. And pests, again, mammals, birds and insects. So, most of these pathogens and pests and parasites, they've been with us for a long time, as long as we've known bees, really, and there are accurate descriptions of these as far back as Aristotle in his agricultural treatises, and in the Roman writers also on agriculture, describing some of the earlier disease symptoms, and we recognize descriptions of wax moth, for instance, and sac brood and paralysis. Then there were some reports by the monks in Ireland in the Dark Ages of great mortality in bees. And then in the Enlightenment era, there was an increase in literature, bee research literature, and practical literature. Then the first description of a true pathogen, sac brood virus, including the evidence that it actually caused the disease it was supposed to cause, a virus. Most of our information, though, and our knowledge from pathogens and parasites comes from research in England in the last 50 years at the Rothamsted Research Centre by Bailey and Ball. And of course, ever since, with help from people in the United States and Australia, significant contributions there and in Europe. But now, of course, and especially recently, there's been a lot of interest in honeybee pathology and in the genetic era as well, the genetics of these diseases. So, first we'll talk a little bit about mites and their viruses, which is a lethal combination. Here's I don't know how many beekeepers there are. I presume there are quite a few beekeepers. So most people beekeepers will be familiar with the varroa mite, what it looks like. Here's a little background. Its original host was the Asian honeybee, Apis cerana, not Apis mellifera, the one that we have here. And they believe that it switched to, adapted to Apis mellifera in around about 1950s, somewhere in the far reaches of Russia, northeastern Asia. And then, maybe with the help of human transport systems such as the railways, it arrived in Europe, and by the 1980s it was pretty much established through most of Europe. The mite reproduces on pupae, but it also feeds sporadically on adults, and there's roughly a 10-to-1 preference for drone brood, so they replicate and reproduce mostly in drone brood. The other significant mite is the tracheal mite, Acarapis woodi. This is what it looks like. Here are some of its features. This one is actually original to Apis mellifera as far as we understand. It only affects adult bees. It infects the trachea. Those are the lungs, so to speak, of the bees. It is transmitted by contact. It doesn't really live very well outside the bee. It really has to be inside. And so close contact is required for transmission. And therefore, symptoms of tracheal mite infestation tend to appear when there is overcrowding in the hive. It's also associated, for the same reason, we think, with winter losses. Then we have viruses. This is what most of the viruses look like under a microscope. However, there's great difference amongst them. We have roughly this number of viruses, depending on how you want to break up the groups. The viruses that are in color, we have determined the genetic sequences. The viruses that are in gray, we still have to do. With this genetic information, we've been able to classify their relationships. And the red viruses are part of a group of viruses called IFLA viruses. The blue viruses belong to the disister viruses. They're very closely related. They have a little different structure, but they're closely related otherwise. And we call them generally picorna-like. And those are these little balls that you see here. That's what they look like under a microscope. But there's a great variety. The other viruses, we have an entirely new family for chronic bee paralysis virus. Some of these viruses new viruses have been recently described in the literature, and it may be possible that some of these are in fact the same as these other viruses that we still have to characterize. And we're working on trying to determine whether that's indeed the case, whether they are the same or whether they're truly distinct viruses. So here are some symptoms. This is sackbrood virus. This is the very characteristic feature that you see when you pull out a larvae. That's what they look like on the frame with their heads turned up a little bit. Another common symptom is paralysis. Paralysis can be caused by a number of different viruses. So we have chronic bee paralysis, acute bee paralysis, slow bee paralysis. IAPV and Kashmir bee virus, Israeli acute paralysis virus and Kashmir bee virus, they're closely related to acute bee paralysis virus. They're sort of in a group together. One of the features of paralysis is that there are many behavioral changes, that's the paralysis, and so bees that are affected are not particularly liked by bees of the same hive. And so they start attacking it and nibbling it until it becomes denuded of hairs and it looks like a black bee. And that was one of the symptoms that Aristotle apparently also picked up. So we have a suspicion that paralysis has been around for a long time. Then we have deformed wing virus. These are the symptoms. And deformed wing virus is the virus that is closely associated with mites nowadays. So next we're going to talk a little bit about transmission. And from a virus perspective, transmission is the most important part of its entire life. It's really what determines its existence. Viruses are entirely dependent on a host for their reproduction. And so viruses only survive by transmitting from an infected host to a non-infected host. It has no life outside it. Successful transmission is therefore at the center of the biology and the pathology, the virulence and the evolution of a virus. Now, infection does not always have to produce a disease, symptoms. Actually, from the perspective of a virus, killing a host before you get yourself transmitted is essentially suicide. So many of the viruses that we're currently discovering in all sorts of plants and animals are what we call now the symptomless viruses. The viruses that we know in honeybees, however, we all know of those because they have caused diseases. So viruses are capable of causing diseases, but they don't necessarily have to. Death of a host or symptoms of a host are only of a benefit to a virus if they enhance transmission. Sneezing is an example for that, for instance, which is also caused by a virus. Sneezing is a symptom that encourages transmission of that particular virus, and therefore it's useful for the virus to develop those symptoms. So there are many different virus transmission routes available for viruses in honeybee systems, and these are some of the ones that we've identified. They can get transmitted by contact and through air amongst adult bees. They can get transmitted through cleaning activities, feces, and trophallaxis. A very important route is through feeding of the larvae. And finally, through cannibalism and cleaning of the cells. As you can see, the adult bees are closely involved in all of these transmissions. This is oral and contact transmission. We have, and by and large, this type of transmission is asymptomatic, generally. It can produce symptoms, but by and large, this kind of transmission does not cause much problem. We also have sexual transmission of diseases, virus diseases in honeybees. Here we have a venereal transmission from drones to the queen. And this is followed then by vertical transmission through the eggs or the egg shells to the eggs. And it's also possible for it to be transmitted directly through the sperm to the eggs. This type of transmission is also, by and large, asymptomatic. And the final transmission is through a vector, in this case, the varroa mite. For those people not familiar with it, the reproduction takes place like this. The mite invades the cell before capping. Then after 2 or 3 days, it starts laying eggs. These develop into young mites, female mites. They get mated, and then out comes the bee with the mites on its back. There is also transmission amongst adult bees of viruses by varroa. There's no evidence that the tracheal mite transmits viruses. Doesn't mean that there is no transmission, but there's no evidence that we've discovered so far. And as I said, through the pupae. And this is the type of transmission that causes symptoms in bees, or the deformed wing virus symptoms at least, and very high titers. And this is a type of transmission that is damaging. Now, as I said, it's always useful to look at transmission from the perspective of the virus. So, from the perspective of the virus, if it gets transmitted during one of these early stages by a mite, and it kills the pupae before the bee emerges, it kills itself. Actually, it also kills the mite, but it's not particularly useful. If Titer is the amount of virus, the concentration of virus, number of virus particles. I'm sorry. So from the virus perspective during this transmission route, it is much smarter, more intelligent, to wait until the bee emerges, because then both an infected bee emerges and mites for further transmission. We'll come back to this a little later. So, what do we know about viruses and transmission so far? And this is a table with the different transmission routes there in blue on the left. We have associations with other pathogens or parasites. Then a sector in the red of the life stages that are infected and cause disease, so it's infection and pathology. And then seasonal incidences of these different viruses. So you can see right on the left that pretty much most of the viruses have an oral route. Question marks, as you can see, there's a lot that we do not know. The pluses means that that we have positively proven that that transmission route exists. Negative ones means that we've positively proven that it does not exist. Question marks, of course, are unknown, and the little squiggles means that some people say yes, some people say no, we're not quite certain. Now, of particular interest, of course, is the varroa mite transmission here, and you can see that that most of these transmission evidences are for the picorna-like viruses. For these, we have no real evidence. What evidence there is tends to be negative. And it's mostly the picorna-type viruses that we're interested in. Notice that sac-brood virus, which is a disease of young larvae before pupation, does not really have much of an interest in varroa transmission. You have associations. Varroa tends to be associated, including with some viruses that it may not directly transmit. When you have varroa infestation in your hive, there's also a direct effect of the mite. The colony gets weaker. Viruses, by definition, are opportunists. They wait for a moment of weakness and then they attack. We all know how this happens with humans. It's the same with bees. And so, some of these associations may be secondary. It's just a virus taking advantage of a weakened hive, but no necessarily direct transmission. And then we have seasonal incidents, they vary. Most of the viruses that we know of cause some sort of disease in adult bees. And as before, in all these transmission routes, the adult bees, they're the ones that are most mobile, they're the ones that are most interactive, they're the ones, the worker bees, that provide the best conditions for transmission, be it oral transmission, through cleaning, through contact, sexual transmission. Sexual transmission, of course, is also a very popular route because with a single transmission event to the queen, you can then instantly infect the rest of the colony through the eggs. And so you see that a good number of these viruses also have a transovarial vertical transmission route and possibly venereal. For the other ones, we don't know yet. Same with the mating flight. Drones fly over large distances to carry a virus, potentially, across large distances to new hives. So sexual transmission we find is a rather popular transmission route. It's also an asymptomatic one because for both of these activities, it's in the interest of the virus not to damage either the drones too much for mating or the queen too much for further transmission. Yes, these are the associations and the fact that most of the viruses have some sort of effect or symptom in the adult bee. So now we go to an example of a collapse of a colony under different circumstances. First of all, in this graph, and this is work by Steve Martin a little while ago, we look at the number of bees in a colony over an entire season, April to April. And here we have a healthy colony. We also plot at the bottom here the age of the bees. So right in spring at top in April, you have a group of very old bees that have overwintered. And they're responsible for starting the new generation in May. And you see a colony developing. The age stays fairly constant throughout until September, when you start getting the production of winter bees. And then once it overwinters, that cluster of bees gets older, doesn't change much, it shrinks a little bit until you get to March and April again, and then the cycle starts over. So now, what happens if we introduce a virus transmitted by Varroa mites? And the virus we chose, or he chose, was acute bee paralysis virus. Acute bee paralysis virus, as the name implies, acute paralysis. It causes paralysis in adults. It also causes rather quick mortality in pupae if it gets infected by injection and other methods. So you start out with the same colony. You can see that throughout the season, this colony is fragmented. It never really develops, and it peters out by September, and it's dead. So that's what happens when a mite transmits a virus that tends to kill quickly. Here we have deformed wing virus, and again, the beekeepers will recognize some features of this. You start again with a small colony in the beginning of spring with old bees, and the colony develops. It's not quite healthy, but it has a reasonable development. It has a smaller winter cluster right at the end, but you know, it manages to get to the end in April sufficiently to be able to start potentially a new colony. So, and the difference between these two scenarios lies really in that pupal stage. So, acute bee paralysis virus, when it gets transmitted, it tends to kill the pupae far more readily and far more quickly before they have chance to emerge. So the colony doesn't develop. Then at colony level, you have a similar type of scenario that because it kills quickly during the season as well, it does not have a chance to survive until the next season, where it can start possibly through splitting, it can start a new colony. So it can survive, and it has another season of chances to transmit. And so both at individual level and at colony level, it is the slow virus that has the smarter strategy for long-term establishment within the bee communities. And this is something that we find not only in this example, but also for some other bee diseases, that the diseases that kill really quickly at individual level very often tend to be not so successful at the colony level. Everything is related to the possibility to get transmitted. At colony level, this transmission from one colony to the next is also through mites, from an uninfested colony, from an infested colony to an uninfested colony. And this transmission by mites tends to happen very often during the robbing season, when weak colonies get robbed out by stronger colonies. And so the stronger colonies take the mites out of the weak colony to the strong colonies. Yeah. So ultimately, however, and the other thing, this is also, this is not just in this particular experiment, which is one season, but for those people with a long memory, this is also the pattern that is played out historically during Varroa invasions into new areas. In the 1980s, deformed wing virus was initially fairly unknown. It was acute bee paralysis virus that was first in Germany and in the Netherlands and in England the one that was most closely associated with Varroa transmission, causing great damage. In the United States, when the mite invaded there in the 1990s, the initial local version of acute bee paralysis virus that was there, which is Kashmir bee virus, was also initially the one that was mostly associated with the bee parasitic mite syndrome and Varroa transmission. And in both continents, over time, as we now know, the main viruses after some period of selection tends to be deformed wing virus that is associated with mite infestation. Ultimately, of course, also deformed wing virus infection and transmission kills a colony, but rather than it having to colony being killed in a single year, it tends to take 2 or 3 seasons. Over successive years, also in the healthy bees, the deformed wing virus load or titer tends to accumulate. You have transmission by Varroa mites that causes symptomatic bees and also high titers. Those bees then also become much more infectious for other transmission routes. So the oral transmission route, which is inefficient, it requires a lot of particles to establish, becomes also more frequent because you have more bees with high titers of virus produced by the Varroa transmission. So there's a mutual enhancement of these different transmission routes. And over a period of 2 years, the colony will also collapse. This is the stage when you see many deformed bees in your hives, and you know that you have to treat. So, again, I'll come back a little bit to this later on. I would like to talk a little bit briefly about another topic of interest, colony collapse disorder, which at its most fundamental is really a case of a mysterious disappearance of adult bees. These are the characteristics of colony collapse disorder. You have an apparently normal, healthy colony, and all of a sudden, within a period of 1 or 2 weeks, you get a nearly complete adult depopulation. The adult population just disappears. There are no dead or diseased bees in or near the hive. So often what you see with diseased colonies, that they have accumulation of dead bees right out in front, you don't see that. And you have a laying queen and large amounts of healthy brood, apparently healthy brood, and a few very young bees around the queen. Secondary characteristics are that you can have both affected colonies and non-affected colonies in the same apiary. And this data is from the United States, and so they found this in 2006 in nearly all the states nationwide, happening pretty much at the same time, which is very curious. There is some evidence that colonies that are depopulated so quickly are less subject to robbing and invasion by pests. And there's also tentative suggestions, no real conclusive proof, that transferring your honey boxes, which is what you do as beekeepers, you try to save your honey, put it on healthy hives, that you may transmit the condition. So,
please proceed, but I heard you have some 10 or 11 pictures left.
Am I too slow?
Yeah, well, no. We were kind of badly organized, so you started too late, but time is limited.
Okay.
Yeah.
I'm sorry. So, the... yeah. So there's a saying that those who cannot remember the past are condemned to repeat it. And there's strong evidence that this particular condition is not at all new. If you look at history, apart from those early events that I talked about in Ireland in the Dark Ages, there have been numerous reports of pretty much identical descriptions throughout history, from the late 19th century to right now, culminating, of course, with the case here in the United States in 2006 and 2009. And so, of course, this precedes pesticides, for instance, mobile phones, all sorts of things. So I'm not sure whether any of those by themselves are the sole explanation for these symptoms. There is also a persistent, for many studies, a persistent association of these symptoms with Nosema infections and one of the acute bee paralysis-like viruses. Kashmir bee virus, Israeli acute paralysis virus, or acute bee paralysis virus. And we don't know exactly how those are involved. However, there are a number of features of this particular syndrome, CCD, colony collapse disorder, that are rather strange for an infectious disease. For instance, you have apparently healthy colonies. You have large amounts of brood. That in itself is a sign of essentially a healthy colony. And all of a sudden the bees disappear. Usually when you have an infectious disease, the colony tends to resist it over a long time. If you saw earlier with the mite-transmitted acute bee paralysis virus, which is one of the viruses associated with CCD, if you get that very lethal transmission, even in that scenario, the colony tends to try to survive for at least 3 or 4, 5, 6 months. The same virus associated with CCD depopulates the entire colony and collapses it in a matter of weeks. It also happens simultaneously over a large geographic area. Again, usually if you have a new pathogen arriving to a continent, it tends to develop slowly, progress through successive stages. It doesn't all happen at the same time. So there are a number of curious features that don't quite match a biological pathogen, but there is a definite association of certain biological pathogens with this phenomenon. How those two concepts have to be combined, nobody really knows just yet. So, impacts, how are we managing? From a beekeeper's perspective, and this is going back to the varroa, of course, you have a slow progression of the varroa mite across stages, across Sweden in this case, and these are the sort of the varroa zones. You have zones where you cannot transmit out of into into uninfested areas. You also have from CCD, again the other example, you have also a beekeeper's perspective. Over here, the interesting part to look at is from 2006 and 2007, when CCD was first described. You had a large percentage of beekeepers that reported the syndrome, and you had large losses associated with those beekeepers reporting CCD. Other beekeepers reported normal losses, and the normal losses were around 16%, which is average. You go 2 years later, in fact, you have a more extensive survey, and the beekeepers identified the causes of death in their colonies as follows. Most of the beekeepers picked out starvation or queen problems. Then you have weather. Mites is the top biological cause of honeybee losses. And CCD has petered down to the bottom, 7% reporting, with still significant percentage losses. But then all of the losses tend to be accumulated. From a regional perspective, and this again comes back to the varroa mite situation, this is from Potts et al. Taking 1985 as a year when pretty much most of Europe was invaded by mite infestations, they looked at the increase and decreases in colonies 20 years before and 20 years after infestation. And you see that there was a large percentage increase in Sweden and Norway during the 1960s and 70s, and a proportional decrease in afterwards. And you have the same, and this may be relevant, you have a similar type of scenario for the number of beekeepers. Again, you have in the 70s and 80s an increase, and in the 90s and the noughts you have a decrease. So these may be very well related, of course. If you look more closely at the situation in Sweden over a long period of records here, you see that there's a huge increase in colonies with a parallel increase in beekeepers, followed by a decrease. And over here in the mid-1980s we have varroa entering as well. This is mostly to show that although varroa will definitely have been a contributing factor in this particular decrease here, there are many other factors that contribute to losses of colonies, such as immigration, such as land use, such as demographics, people moving into cities, immigration, things like that, that should be factored into these equations. And so proportional increases may not give you a complete representation of what happens. Solutions looking into the future. The first slide here is what we do currently to control the varroa mite. It's imperative that you control the varroa mite to limit its damage through virus transmission. We have the synthetic acaricides. By and large, these are very effective, but there is evidence of resistance. There's residue development. Currently in Sweden, you need a veterinary license for some of these. You can only apply them before or after the honey season. But they are effective and they're easy to apply, not necessarily cheap. You have the natural acaricides, or the organic acaricides. Those are mostly organic acids, formic, oxalic, and lactic acid. And the organic oils, mostly thymol. They work through vaporization, which may be a part of the reason why they're so difficult to implement. You can apply them at any time, but usually they're applied after the honey harvest. We have formic acid. That's the only one that is effective in pupae as well as on adults, so it affects mites everywhere in the colony. There's a question with these of brood and bee toxicity. Formic acid tends to be very corrosive, particularly to open brood. You have an aggression in bees, and there's a temperature timing effect. It's difficult to apply. You need to be very careful. These are caustic materials. There's a temperature effect. Evaporation is dependent on temperature, number of bees flying in and out. So they're not as easy as just putting in a strip and coming back 6 weeks later and taking it out. Then you have biomechanical methods, drone comb removal and screen bottom boards. These can be applied throughout the season. Mites have a strong preference for drone comb and drone brood. And so by removing combs of drone, you can remove a large number of mites. Same with the screen bottom. That is basically mites falling through the bottom and not being able to enter into the colony. You can apply those at any time. The effects are moderate or marginal. Neither by themselves will manage to keep your colonies completely varroa free. At some point, usually you will have to have some sort of additional treatment. But both are very useful in an integrated pest management system. And then you have genetic. We know there's genetic variation for varroa mite tolerance. Breeding efforts have been rather disappointing. There's no real resistant honeybee line, despite very, very large efforts.
Now we have to close soon.
And then finally, I'd like to thank you, and my apologies for mistiming it all.
I'm sorry. I'm so sorry.
No, that's my fault.